Well, it's a pleasure to be here. I was last year, I think it was 2002, and ah, let's try and okay, oh good. I'm very short, so that's better. That's much better. Let's try and get my first slide up there. Can you do that? Good. Okay. First of all, thank you very much for this occasion. Thank you for all the nice things that have been said. It's really a pleasure to be here again. When I was last here, I was still running. I'd set up Cancer Research UK, and which is the major funding agency for cancer in the UK. But since then, I have not been directly involved in a cancer research institute, although the CRIC is significantly funded by Cancer Research UK, but it covers all aspects of biological and biomedical sciences. I've worked all my life on the cell cycle, and you could say I'm not very imaginative for doing it. And it's now for 50 years with the Nobel Prize exactly in the middle. I've worked on it because it is such an important process, fundamental to life. It's the underpins the growth, reproduction of all living things. And in a sense, has to have been the first proper developmental pathway in a eukaryote in the origin of life, because the first life would have been single-celled, and the cell cycle is the development of that single cell from 1 to 2. So it is like developmental biology. You need to understand temporal and spatial order, and I focused on temporal order. But unlike development, it is something that is absolutely common to all eukaryotic cells. There's, of course, overlap with development, but the cell cycle is universal, and that's why I've worked on it. In this talk, I'm going to give you new perspectives on the role of the cyclin-dependent kinases that were partially discovered in my lab, also in Lee Hartwell, Tim Hunt, who shared the Nobel Prize. And we had a story 25 years ago, which has got into the textbooks, but I actually want to change it today. I want to give you a different, it's not wrong, it's just not fully developed, and I want to develop it further. And I want to say some things that I hope will guide you. I work on a very simple system, yeast, because it's simple to do experiments, and you can do very effective experiments. But it's very important to see whether some of the things that I'm going to say apply to more interesting cells that we have that make us up. Okay, so that's a start. This is a mammalian cell undergoing cell division. It's the end of the cell cycle. This is I drew this about 35 years ago. I still use it. This divides the cell cycle up into its developmental sequence with the major events, which are, of course, S phase, when the chromosomes replicate, mitosis, or M phase, when those replicated chromosomes segregate into the two newly divided cells, with processes occurring in G1 and G2 that are important to lead to those major events, and the other things that happen in the cell cycle as well. I won't talk about those other things today, but we shouldn't actually forget those. Now, most of the work that I've done has been carried out, that I'm going to talk about today, except for the first part of the beginning, has been carried out in the Francis Crick Institute, which, as you heard, I'm a director, and I set it up. This is the logo. It's a dynamic logo. Look at that. Watch it. Bang. This is the most interesting slide in my talk, by the way. I said I wanted a logo that didn't look like any university logo or any research institution, and that's what we have. We can change the colors according to our mood, and so it's a very, certainly a very dynamic one. And this is it, which some of you have visited, and it's a large institute in the centre of London, next to St Pancras Station, has 1,500 scientists in it, no departments or divisions actually, a little bit anarchic, which is appropriate for a discovery research institute. And one major reason I'm here is to try and forge stronger links between the Francis Crick Institute and great institutions of biological and biomedical science in India, which is a reason I came here, so that we can have links. Particularly, I'm thinking of young researchers who can get experience in the Crick that can come from India. And I stepped down from being director of it in a few months, actually, but will take up the Royal Society presidency, and a major task there will be also to open up stronger relations between India and the UK. India's tended to be looking at the US for the last decades. That is, of course, something that's looking a little bit more complicated, which is, of course, an understatement, but a little bit complicated now, and we really should establish other connections as well. I'm going to start by giving what one might say the textbook view of CDKs and the regulation of the cell cycle, so that you can compare with how it's different. This I stole from some textbook, and it goes a bit like this. Cyclin-dependent kinases consist actually of three components. The CDK, that's the protein kinase, a cyclin activating component, and a third component, CKS it's called, which is actually beginning to become interesting in cancer. And given that, I've actually got a whole study going on in that, but I'm not going to talk about that today. I'm just going to talk about the first two elements, CDK and cyclin. In many organisms, including ourselves, there is a range of different CDKs, 4, 5, 6, even more actually, but they get more diverged. But 4, 5, 6 that have been, 4 to 6 that have been implicated in the cell cycle, and also a similar family of cyclins called B cyclins, A cyclins, D, E, and so on. So the combinatorial series is quite significant. And generally, how that's been interpreted is schematized in a simplified way here. And it goes a bit like this. I say it goes a bit like this because, frankly, it isn't very clearly evidenced by the work that's been done. So it sort of got into consciousness without actually having such a strong evidence base. It goes like this. There are a range of different CDK cyclin complexes. They have different substrate specificities. That is, they phosphorylate different sets of substrates. And the phosphorylation of the different substrates bring about the different events of the cell cycle. So in a simplified version, there will be G1S and S CDKs that pop up at the G1S and S time in the cell cycle and phosphorylates proteins, important for the G1 transition and S phase. There will be other events that occur in G2, and then, of course, the major mitotic CDK, a different combination of cyclin and kinase that comes up at mitosis to bring about the events of mitosis, followed by degradation of the cyclins discovered by my friend Tim Hunt, working in, by the way, marine eggs, just to know where these discoveries come from. The cyclin is then degraded and cells exit M phase. Now, I think this, for those of you who did this in undergraduates, that sounds about right. I mean, I think that's what, and this is, I'm going to suggest that we haven't quite got it right, okay? Now, I started on this problem for the reason of the next two slides. Now, here we have cyclin CDK, and around it there's lots of arrows, which are all the regulation that's been studied by many, many groups. And the only thing you need to take from this, because you don't have to read any words, is there's a lot of it. There's a lot of regulation, and it's complex. And when we model this, we get something like this, okay? Lots and lots of elements that are all connected with these arrows and so on, as you can see here. And I hope it's too small to read. I don't want you to read it. I want you to look at it and get mildly depressed. And the reason I'm saying that is, does it really tell us anything? You know, it's a little like the underground map in London, okay? And if you want to go from, you know, Hammersmith to Barons Court, it might be useful. But this isn't very useful. It is a collection of a naming of the parts with spurious links, often, just to give a complete picture. So I got a bit depressed by it, even if you didn't, because I noticed you laughed, okay? And I got depressed and thought we should try and take the following approach. We should try and simplify the system. It's quite simple, it's fairly simple in yeast in the first place, but let's try and make it even simpler, because then we can focus on if it still works, even if it's simplified, it focuses our attention on what is important. And then maybe if we're focused on what's important, we can establish principles that will help us look at the much more complex situation that we have in, for example, our own cells. So that was the objective. And a postdoc came to my lab, Damien Kadrusa, and we started to simplify the system by just fusing CDK and cyclin. Curiously, it had never been done before, but the objective was just to get rid of the controls that find the two proteins, puts them together, and makes them stable. I just thought it might make it a little bit simpler. Now, this was in fission yeast, and this is the problem. As I said, it's not so complex. There's only one CDK, first of all, encoded by the CDC2 gene, called CDK1 in human cells. There are 4 cyclins, PUC1, SIG1, SIG2, CDC13. You can read this slide now, that's allowed. And you'll see as you go around the cell cycle, these are, the traditional view was PUC1 is involved in G1, SIG1, SIG2 S phase, and so on, CDC13 on onset of mitosis. So that was the fission yeast cell cycle, what Damien was looking at. And he was fusing CDC13, CDK1. Played around with that, got it to work, and this is the result. This is the first time you've seen a fission yeast cell. It's really like a sausage, you know, just grows, elongates like a sausage, undergoes mitosis, divides in the middle, and splits. So you see wild-type cells there. And that means, by the way, it's very useful, the length of the cell tells you where you are in the cell cycle, which is actually the reason my boss back in Edinburgh, Murdoch Mitchison, started to work on this particular problem. The middle panel there shows the CDC13 with a linker, that's L, fused to CDC2, and Damien deleted the endogenous CDC2 and CDC13, and you see that works perfectly well. There's a Western blot there to confirm that the fusion is there. But Damien, who was bold, went through and then deleted SIG1, SIG2, and PUC1. So the whole cell cycle, and it works perfectly well. So the whole cell cycle is being driven by one cyclin-dependent kinase. Now, you can immediately see this blows a hole completely into this qualitative model of different CDKs with different qualitative substrate specificities, and we're forced to think of something else. We also looked at meiosis, where there's 2 further cyclins, deleted those as well, so all 6 are now deleted, can be driven by a single CDK. So both mitotic and meiotic cycle are functional. The model we came up with is here. On the left is the conventional picture of the cell cycle, and you can see the color, the depth of redness. It reflects the level of CDK activity. And in G1, the activity is very low. You get a small amount of activity, and you get S phase. You get more activity, and you block S phase, We showed that, and that's the reason why actually you don't get 2 rounds of replication in the cell cycle. It's actively inhibited. And then when you get a lot of activity, you get mitosis. And I've rolled out the cycle on the at the right-hand side, and it makes some predictions that we can test. And the way we can test them, then I'll tell you the predictions, is that we can mutate the CDK1, the kinase, and mutate the ATP binding site. This was invented by Shockat. I sometimes call it, I use his name as a verb, we Shockatize the kinase. And what it does is it enlarges the ATP binding site. So a bulky ATP analog can now bind and inhibit that kinase, but not any other kinase in the cell, at least in principle, as long as you have to do controls and so on for it. Now, that's usually used in a sort of single way. You add a lot of inhibitor and you block the kinase, okay, and it allows you to interpret. But the reason we did it was actually to add a range of concentrations, so we titrate in different levels. Because what we are proposing here is a very simple model, not a qualitative one, but a quantitative one. That is, the level of CDK rises through the cell cycle. In a wild type, that level is contributed by a range of different CDKs. And at low activity, you get S phase. At high activity, you get mitosis and all sorts of things in between. Now, this could not be a simpler model. So it's completely different to the complexities. We're not looking at how we control timing and so on. It's very, very simple. It's like a rheostat, really. You're winding up the rheostat, and as you put more electricity in, you get out different outcomes. Now, the Shockat inhibitor allows us to test that. Because if you look at the right-hand slide, if you need a lot of activity to go into mitosis, you'll only need a small amount of inhibitor to block it. If you only need a low amount of activity to go into S phase, you will need a lot of inhibitor. And if, strictly speaking, this model's true, if you're in the middle there, in that pink level, where we're blocking S phase, if we now add inhibitor and force the kinase activity down, and then wash it out again, what we, in principle, should be able to do is to redirect the cell into G1 and then allow it to endo-reduplicate. All those experiments were done and worked. I'll just quickly go through them. This is what happens if we synchronize in G2. We release cells so they go into mitosis. The DMSO control is the black line, as you can see. As you have increasing concentration of inhibitor, it delays and gets lower in peak, and then eventually at 0.3 micromolar, or somewhere between 0.1 and 0.3, no mitosis. I just want you to remember 0.3 of a micromolar, between 0.1 and 0.3. Next, we looked at G1S, and this is shown here. This time we used FACS analysis to ask whether cells are in G1, G2, and so on. And so are they 1C or 2C? Now, fission yeast has a very short G1, and under these conditions, we don't even see it pop up in the experiment. So the DMSO, the black squares, are G1 cells. If we add 1 micromolar, you see we have a 30-minute delay on S phase, so G1 is extended by 30 minutes, you see that. 2.5 is extended to 60 minutes, 5 to 90 minutes. If we put 10 in, and we can't go above 10 because it starts hitting other kinases, we see about 120-minute delay. Now, what I just told you is to block S phase, we needed 0.1 to 0.3. To block mitosis, we needed 0.1 to 0.3. But to block mitosis, we need something like 5 to 10. So it is 30 times more. I don't quite know how it relates to activity, but for sure, there's more activity present when you've got 0.1 of a micromolar than 5 micromolar. So that sort of fits. The fun experiment in this one was to get cells to re-replicate, just shown here, again, FACS analyses. We block cells in G2. We now hit it with a high concentration, so the cell now thinks it's in G1. We wash it out, and then it endo-reduplicates. Can you see the peak on the right-hand one undergoing undergoing DNA synthesis. Tells us a couple of other things. It suggests there's no arrow of time in the cell cycle. It means you can slip backwards and forwards. And the reason you can do that is because the position in the cell cycle is identified by the cell by the extent of the CDK activity. So when you have low activity, the cell is in one place. When you have high activity, it's in another place. So you can shift where you are by changing the concentration we have there. Is this all just something to do with fission yeast? Okay. Well, you could say that might be true, but I'd be careful about saying it because mostly what we discovered in fission yeast did apply to other cells. So I'll just give you some. I did some literature search just to see. Quickly, because I want to get onto the newer stuff. There's 9 different cyclins in bug-in yeast. If you can delete 7 of them and undergo a normal cell cycle completely, you can delete 8 of them, so you only got 1, and the cell will undergo S phase and mitosis, but not budding. So that's exactly the same as fission yeast. If we look at Xenopus, it was always thought that the G1S CDK was not interchangeable with the mitotic one, and so the mitotic CDK couldn't drive S phase. Then it was realized that the majority of the kinase was in the cytoplasm, when of course DNA replication occurs in the nucleus, which we're going to come to a bit later. And so if you put an NLS, a nuclear locating sequence, onto the CDK, it went into the nucleus and drove S phase. So that didn't hold up. If we now look at mammalian cells, the word plastic is introduced a lot. Whenever you read the word plasticity in a paper, get suspicious, okay? Because what it really means is that it didn't actually work out as we expected. And so we're going to say it's plastic, and then we can move on and not worry about it very much, okay? And I think one should worry about it, to be quite honest. And why I say that is that the only CDK in tissue culture that is absolutely essential is CDK1. You can get rid of all the others and it can be substituted. It does vary a bit from tissue culture to tissue culture. And the only cyclin that's absolutely essential is B. They haven't done the same sort of elaborate experiments we've described here, but you can delete each one individually. And what I'm going to say is that I think this part, then I'm going to move on to more current things. There is a core principle for understanding CDK control of the cell cycle, the rheostat model. That's the core principle, but there are peripheral controls relevant for multicellular organisms like our cells or a mouse, which of course have got to respond to being in a tissue, being in development, being in an organ, being in an organism, which has layered on top of it lots of other controls, which are likely in tissue culture to be still hanging around, which shows why there's so much variation in different experiments, which has really confused us. And I say this so clearly because I think we should re-examine that data in the context of what I just described. All the experiments now could be done like this. Now, it could be done in mammalian cells. Now, we went on in more recent work, which has all been done in the Crick in my lab by mostly graduate students. This one is Matthew Swaffer on the right. On the left, somebody who worked in the proteomics facility because it involved mass spec. By the way, 3 of the pieces of work I'm going to talk about were all led by Indian scientists, 2 graduate students, 1 postdoc. And we reasoned that if CDK activity was rising as you go through the cell cycle, that instead of getting peaks at S phase and peaks in middle of G2 and so on, probably the substrates, or many of them, would just continue to be phosphorylated. The qualitative model suggests that peaks, and the other quantitative one not. So what we did is, or Matthew did, was to make synchronous cultures, take samples, you do mass spec phosphoproteomic studies, and you see the answer here. Light blue are S-phase substrates, because we know which ones they are. You can see it goes up at S-phase, but it doesn't go down again. It continues to go up as the activity increases. So there is no sort of peak, it's actually continuous. Mitosis requires a higher level, so nothing happens till the end of the cycle, then that goes up. Then we looked at the amount of the shockat inhibitor, NMPP1 here, that was needed to inhibit S-phase substrates and M-phase substrates. And you can see here, to inhibit mitotic substrates, you only need maybe 100 to 300 nanomolar. To inhibit the S-phase substrates, just as we'd shown previously, it's something like 5 micromolar. So that explains the biology, looking at the substrates. We could also use this methodology to do in vivo protein kinase assays. Normally, what we would do with a protein kinase to assess what a protein kinase activity was in a cell, normally what you'd do is break the cells open, set up an in vitro assay, kinase in it, you'd add ATP and a substrate, and look at the rate of phosphorylation of the substrate. Agreed? We can actually do that in vivo, because if we have now the shockat version, we can take cells, inhibit the kinase, the turnover rate is 2 or 3 minutes, dephosphorylate all the substrates, and the experiment at this time we were looking about 250 that we just assayed all in parallel. So you then wash out the inhibitor, and it's like adding kinase back. Then you can do a time course and measure the activity on 250 substrates. I mean, this is, of course, technology that could be used for any protein kinase, should you want to. The left-hand slide shows that the activity in a G1 cell is pathetically low to an M-phase cell in vivo, and it continuously increases as you go through the cycle. On the right-hand slide, we divided up the data from this according to whether the substrates were late in the cycle, in the middle, or at the beginning. And as you can see, substrates at the beginning were much more readily phosphorylated than substrates at the end. So this is now the summary of this work. We have the cell cycle on the left being driven by a single CDK. We have the pattern of early, mid, and late substrates as you go synchronously through, and you see they come up at different times. And on the right-hand side, you see we have CDK activity going on the horizontal axis, increasing in phosphorylating G1S substrates to get you G1 and then S and then G2M substrates. So the basis of temporal order through the cell cycle is this quantitative increase. Now, there was one problem, and it bugged me for a couple of years. And the problem was that if what we're saying was true, then the G1S, we got the cell cycle driven by the G2M CDK. Why didn't the G1S undergo mitosis? Which it didn't. And we tried very hard to get it. And I'd sort of given up. And then my next Indian graduate student, Saz Basu here, who was very persistent. He'd been trained at Oxford, actually, and came over. And he devised a way of investigating it and solved it, okay? And I have to tell you, I said, this won't work, you're wasting your time. And he ignored me, quite often the case, and quite a wise thing to do, quite often, and did it. And what he did is he set up a system, I don't really want to explain it, where he got rid of all these CDKs, and under a regulatable promoter, either made a CDC13, CDC2 fusion protein, or a SIG2 CDC2, so G1S CDK, G2M CDK. And if you look at B, when he made the dark blue is the G2M CDK, works perfectly. You see that peak with mitosis. The G1S, 0, okay? Beautifully shown what we'd shown crudely before. But he looked at all the potential inhibitors of CDK activity, it's on the left here, And we could genetically manipulate them quite easily in fission yeast. And he found one of them, phosphatase PP1. If he deleted that, we got a partial rescue. You can see that on the right. So that was encouraging. And then he looked up where was PP1 located in the cell, where is CDK? And we found that PP1 and CDK were located in what we call the spindle pole body. The spindle pole body is the centrosome in yeast, the centrosome. And an ex-graduate student of mine had shown, years after he left me, that PP1 was kept in the centrosome by a scaffold protein called CUT12. And he also showed that if you constructed certain phosphorylation mutants in CUT12, the PP1 was ejected from the spindle pole body. So what Saz speculated was maybe that's where the problem is, that we need PP1 out of the way of the centrosome. And if you look at the bottom, he was exactly right, and I was quite wrong to discourage him. When PP1 is ejected from the centrosome, there's still CDK there, then what happens is you get complete rescue of mitosis, okay? So now we've shown G1S CDK can bring about G2M as well. So I thought we've done quite enough on that, except for one more experiment. And that is, we could use the way that, that Saz had set it up, where he had cells with just the G1S CDK and the G2M CDK. We could look in vivo at the substrate specificities. I mean, because we could, we could, in this experiment, we assay 276 phosphosites. Okay, 276. Look at this, it's remarkable. These are measuring the substrate specificity of CDKs that many think are very different. 180 of those 276, you look at the dark blue and the light blue, are essentially identical. 180. The two middle ones, 31 and 29, totaling 60, the G1S CDK is actually more effective against mitotic substrates than the mitotic one. So it simply is even counterintuitive. It's only the 36 at the end where there is a difference, and where SIG2 CDC2 is reduced the clustering the clustering approach produced the plot that you can see here. And of those, where half are down, only 12 of them are less than 1/3. Only 12. So around 5%, 6% of the substrates show some difference. Now, most people just don't look at in vivo substrate specificity. We're assaying huge numbers here. But the conclusion is that the substrate specificity of the G1S CDK and the G2M are almost identical. And that's where I'm going to draw a line. Now I think somebody should do some of these things in more complicated cells to see if it applies. And now I'm going to move to the last part of my talk, which is done by my Indian postdoc called Nitin Kapadia. I haven't chosen this because of my audience, by the way. This is exactly what I'm talking about. Now, Nitin came and reanalyzed Matthew's data, Matthew Swaffer. I showed a picture of him, and to look at the pattern of phosphorylation of proteins at the G2M transition in different compartments within the cell. And he was using just the GO analysis, are they in the nucleus, cytoplasm, and so on. And what you see here is that, with the exception of the green line, all the others, which are cytoplasm, the periphery of the cytoplasm, that's on the edge of the cell, nuclear envelope, the centrosome, are all clustered together. But the nuclear is about 15 minutes advanced. Now, we were pretty excited by this because I haven't perhaps emphasized this quite enough, but the CDK at G2 mitosis, when it's activated, is really the pacemaker for progression through the cycle, because if you have more CDK activity or less, you will go, if you have more, you'll go through it faster, and if you have less, you will go through it slower. And this suggests that we maybe shouldn't be looking at the overall CDK activity in the cell, but maybe there was a pacemaker component that was in the nucleus. And the rest of my talk will be establishing that and elaborating a little bit more upon it. Now, what we would, if this was correct, what of course we would have to do is measure CDK activity in the nucleus and in the cytoplasm. Now, you can do that with single cell CDK sensors, okay? And the principle there should be on the next slide. Yeah, it's like this. So we'd already devised, Nitin didn't do this, a kinase assay before we were thinking about compartments, which assayed CDK activity in the cytoplasm. And what would happen is that you'd make a substrate of CDK colored, and when it's phosphorylated, we choose a substrate, it changes location in the cell. And if you make it colored when it's normally in the cytoplasm, it's uncolored, but it's, well, look at it here, actually, and then I'll tell you about it. If it's in the nucleus and it's unphosphorylated, it remains in the nucleus, so the nucleus is colored. When it becomes phosphorylated, it's ejected from the nucleus and in the cytoplasm. So you can measure the ratio between nucleus and cytoplasm to determine what the level of activity is in the nucleus. And the reverse can be used for the cytoplasm with a different sensor. Did that sort of make sense, more or less? Good. So he made sensors that could be used to assay CDK activity in the nucleus and CDK activity in the cytoplasm. The reason this is important is the nucleus only occupies 8% of the cytoplasm. And so when you do normal traditional biochemical assays, most of the material you're getting out of the cell, 92%, may be in the wrong place for where you should be looking at it. Okay, so this is this was a nuclear, what you see in red there is a single cell. We did, of course, many cells which showed similar patterns. This is nuclear activity, and you can see to a first approximation, it increases a bit at the beginning of the cycle when it undergoes S phase, continues to increase as you go through the cycle, just as we predict. And then there's a sudden jump, and you go into mitosis. If we now look at that sensor and also the cytoplasmic activity in the same cell, and we can do that by making the sensors different colors, what you see there is that the nuclear CDK goes up about 10 minutes before the cytoplasmic one, which is in green. You can see as it does exactly what we saw before, it gradually creeps up, and then, you know, we've actually been conservative. You could say it goes up a bit earlier in actual fact, but I wanted to be conservative on it. So that strongly suggested that, that indeed CDK activity was going up in the nucleus first. Unexpected, completely unexpected. And we looked at a number of cells here, and you can see on the right the time of the delay that we actually observed. Now, Nitin then went on, again looking at colored proteins, to look at changes in the amounts of protein, in particular cyclin. Okay, CDC13 shown here. Now look at the whole cell, that's the top one, the green. And the sense of what you get is that it's increasing through the whole cell, goes into mitosis, which is set by those dashed lines, and then it's broken down as you exit mitosis. But if you now look at the level in the nucleus and the cytoplasm separately, which of course you can for single cells, you can see it's a bit more subtle. What happens when the cell goes into mitosis is there is an export of CDK out of the nucleus, and look at the blue, into the cytoplasm. So there's a subtle shift of about 25% of the cyclin. It's exaggerated on the right just to show that more clearly. We looked at CDC2, and we saw essentially the same thing. This is the level of CDC2 and CDK1. And you can see that at mitosis, it drops coming out of the nucleus and increases as you go into the into the cytoplasm. Now, we speculated that the rise of CDK activity in the nucleus made the nuclear membrane more permeable. I'll remind you that yeasts have a closed mitosis. The nuclear envelope doesn't break down. But if the nuclear envelope is highly permeabilized, then that is actually like an open mitosis, only the envelope is broken down. So I wondered if it was an analog of that. And what was happening was that activation of the CDK in the nucleus promoted the transfer of the CDK from the nucleus to the cytoplasm by having an influence on the nuclear envelope. And once some active material was in, CDK was in the cytoplasm, because there's a positive feedback control, which I'm going to explain in a moment, then that would activate all the inactive CDK that was in the cytoplasm. So the level of CDK activity in the cytoplasm is a slave on the pacemaker CDK in the nucleus. Okay? Hope this isn't getting too involved, but that's where we are. Now, luckily, there's a positive feedback control. WEE1 and CDC25, which were discovered in my lab years ago, in the 70s, in fact, we now know that WEE1 phosphorylates a tyrosine 15 on the CDC2, CDK1, and that inhibits the activity. And it's activated by a phosphatase, CDC25, that takes it off. More importantly, we didn't discover this, it was discovered in Xenopus. The active CDK phosphorylates CDC25, activates the phosphatase, and so now you get a positive feedback. If you have some positive CDK here, and that is what's thought to call that jump of activity at the end of mitosis. So we looked at this situation again, and this is sort of background for it, and looked at the timing of what was going on. And here you see, just to get the nuclear export related to the activation in the nucleus, in red, you see here the sudden jump up of activity within the nucleus. And what you see is that it's correlated very precisely to when there's export. So that's the sort of first part of it, export into the cytoplasm. And this is now looking at CDK activity in red in the cytoplasm. And what you can see here is immediately after you get nuclear export, you activate CDK activity in the cytoplasm. Also fits with where we are. So we have the model that we think that CDK is activated first in the nucleus. It permeabilized the membrane. Some part of it is now exported, and that activates the cytoplasm. So you have two domains, a regulatory domain in the nucleus, a regulatory domain in the cytoplasm. And you probably therefore have nuclear events for mitosis, and cytoplasmic ones are controlled in different ways, and we're testing that. But there's something else we noticed. We noticed that the level of cyclin in the cytoplasm and the level of cyclin in the nucleus when the CDK was activated was actually really quite different, the concentration. And you'll see that it is far higher. You need far more cyclin in the nucleus to activate than you do in the cytoplasm. And that made us think that maybe it was more tightly regulated in the nucleus, and the regulation in the cytoplasm was a bit sloppier. So we did some phase plots. Now, you may not be so familiar with phase plots. They're done in vitro, usually, in biochemical assays. These are the first in vivo ones that I'm aware of. But if you look at the left-hand one, on the horizontal axis, we're plotting increase in the cyclin, CDC13, as you're going through the cycle. And we are measuring CDK activity on the vertical axis. And what you see is that as the cyclin's accumulating, it goes up. Then at nuclear activation, there's a sudden jump. Then there's nuclear export, so it goes down a bit, but you're still going up in activity. and then it's degraded as you go down the other side at the end of mitosis. And what you can see if you look at the blue, which you hardly notice, is what it looks like in the cytoplasm. So it's much less tightly regulated and at a lower level. This time on the right, I've done a range of cells to show you that the behaviors are similar. Then we asked the question, well, what is explaining this quite nice phase plot like this? We guessed it might be tyrosine phosphorylation, tyrosine 15. So we mutated it to phenylalanine, so it can't be phosphorylated. And now you see that phase plot collapses. So the reason for the strength of the control is in part due to tyrosine phosphorylation. We can now look a bit more at cytoplasm. So if you look at the top left, everything's scrunched together. We can pull it out, which is below it, and you can see there is sort of some hysteresis and so on going on. It's weaker. And what hysteresis there is that pulls these things apart is completely eliminated on the right there when we look at a phenylalanine mutant that can't be phosphorylated. So that makes us think we've got a much stronger regulation in the nucleus than we have in the cytoplasm. Now, we were interested in one other thing. Could we get better evidence that the nuclear export mattered? It's correlated, but can we get better evidence? And we could, because there was a mutant in cycling called the hydrophobic patch mutant. You don't have to worry about HPM. And the HPM mutant on cyclin didn't allow CDK to locate to the spindle pole body. That's been shown for some years and been known in mammalian cells as well. The same thing happens in mammalian cells. So we wondered if it wasn't actually directly to do with relationship with the centrosome, but maybe that HPM mutant could not be exported out of the nucleus. This was a wild guess, which turned out to be true. What we see here is 20-30 cells looking at CDC13 HPM. It goes up through the cycle. It should go down at about 200 minutes, but it doesn't. It remains constant. And it never gets into the cytoplasm. And if we now go back, I think, to... Oh, no, gone the wrong way. What you see on the left is the nuclear CDK activity, which is activated in this mutant. Look to the right, there's no activation. So if the protein cannot get out because of the HPM mutant, it can't activate the CDK activity in the cytoplasm, it doesn't get activated. That shows the dependency on the pacemaker enzyme in the nucleus is the dependency of the cytoplasmic activation upon that. So this is my summary. And then I'm going to finish with a 2-slide joke experiment. Is that okay? Okay. This is the summary. Not only do we think that the main principle for cell cycle control is in fact a single CDK, and we're working into it to see whether a range of CDKs can contribute to that activity, but as I said, it could be more complicated in a multicellular organism. But what we're saying is that for the G2M transition, all the control is in the nucleus. Now, that makes complete sense. We should have thought of it before. Because what is the most dangerous thing for a mitotic cell? It is to go into mitosis if it hasn't finished DNA replication, or if the DNA is damaged and not repaired. Where is the DNA? It's in the nucleus. So having a domain of regulation, which is very strong because of the phase plots, is perfectly placing the CDK regulatory pacemaker system right next to the chromatin and the DNA. So we're now investigating chromatin as a sort of platform for the regulation and how cells work. When we get activation by phosphorylation of, I think, some membrane proteins in the nucleus, or possibly the pores, then we get export, which activates the much weakerly controlled cytoplasmic CDK shown on the left, which is a slave on the pathfinder. And this is, we will now investigate this in the coming years. I bet you money that the same thing's happening in mammalian cells. I just bet you, it just smells right, I have to say. It just smells right. Now, I have a tradition in my lab of training junior technicians and, you know, school leavers or undergraduates who wouldn't be doing a PhD. And they work directly for me, actually, poor things. But they work directly for me, and I do the training, or I mean, not how to pipette and so on, that's done by others, but to do the experiments, okay, and develop them. And this is Sophie, who's from Ethiopia, actually, and this is what I call my Jurassic Park experiment, okay? You'll be familiar with the film. Now, you remember they got DNA out of a bit of amber from some insect or bitten the dinosaur, I think. So what we did is paired up with Bill Wickstead, who was a bioinformatics evolutionary biologist, and I asked him to predict the sequences of CDK1, cyclin, and CKS at various times in the evolution of eukaryotes. And I was interested in two particular transitions, the ophicecont, which is the one on the right, when fungi diverged from the metazoa, which is thought to have occurred about 1.3 billion years ago. Just to put that in perspective, dinosaurs went extinct at 65 million years ago, so it's pathetic compared with what we're doing here. And the origin of eukaryotic life, 1.6 billion, 2 billion, big distances, okay? Now, we're halfway through this project, but the CKS, I haven't talked about CKS, we have created the origin of eukaryotic life, and it works. Oh, I didn't say. We predict the sequence, then we synthesize it, so we make it, and ask, does it work in a modern yeast? Okay, and we've shown that the CKS that's predicted for the origin of eukaryotic life works. Amazing, yeah, it works. If we look at CDK1, we've got it back 1.3 billion, but we've not managed to get it back 2 billion yet. I've tried 4 variants and none of them have worked. But now we're creating a strain which is growing only with CKS from 2 billion years ago, just in case it's the interaction with that matters, and then we'll repeat it. Haven't tried cyclin yet because it's more difficult to predict, and I thought if we should see whether we could get this to work. This is the experiment, the first one we did. On the left, you've got a temperature-sensitive CDC2 mutant. On the right, it's growing with the ophicecont. Perfect rescue of the defect. We've repeated all this having deleted CDC2, so there's no question that there's no activity. Well, today, this is crick from an aeroplane, which was taking photographs. It's my dreamy crick sort of slide. What I'd like you to take home is think about quantitative models for CDK rather than simply qualitative. I think that the situation in our cells, which you'll be most interested in, will be more complex. But if we understand the principles, we'll understand the complexity of cell cycle control better if we actually learn from the simpler systems. And it shows that there's still things to be learnt with this, and we shouldn't just rest on our laurels when something gets into the textbook. We should challenge it and see where we can go. So thank you for listening to me, and now you'll see the logo will disappear because it's the end of the talk. Thank you very much. Now, I really like questions, but I'm a bit deaf. So I'm going to ask our chair if they can help me if I don't quite get it. Wait for a microphone if you can, though.
Yes, Surab.
One at a time.
way of inhibiting CDK function while it's in the nucleus and keeping it actually regulated or something else?
I think that there's multiple controls, but cycling is clearly important. We need a certain level of cycling to break through. The second control is tyrosine 15 phosphorylation. Both, of course, are operative in mammalian cells. CKS does have a role, which I haven't discussed here, on some, about 200 phosphocytes. But I don't think that's the principal regulator there. It's moving it a whole step in a different place. Okay?
And the second question is the export active? For example, like with B-type cyclins in mammalian cells? Or is it
passive? Yeah.
It is passive?
No, I'm sorry, I was finishing your question.
Sorry.
I don't know, but I put a student now on looking at exactly that question to identify transport out of the nucleus and import and export as you go through the cell cycle. Is there a transition? Does it matter on molecular weight? And so we could actually define that. And we can use FRAP to actually measure import and export. We've got preliminary results, but they don't mean anything yet. So it's a very important question and introduces a whole new level of control. You know, the import matters.
Yeah.
And when we did a genome-wide survey to identify genes that regulated onset of mitosis, we got about 25 different genes. 7 of them were involved in nuclear transport. We did that a couple of years ago. I couldn't explain it. Now we absolutely can.
So if you actually poison export, do you stop? Well, you should stop cycle, but will you or promote export?
Well, I'm a bit reluctant to poison because you hit everything, but we can. I told you we had these nuclear poor genes which, when deleted, will influence onset of mitosis. So we can look at transport in those cells which are viable but actually alter when they go into mitosis. So we will be able to examine that. Thank you.
Any other questions?
Yes, there's one here. Is the mic on, actually? Yes, it is.
So CDK level is quite up, you said, in G2M.
Yeah.
But, so why it is not phosphorylating S-phase substrates?
You mean in the nucleus?
Yeah.
I didn't say anything about S-phase here, because the reality is we've not looked at it. But now, given this, I want to ask if the nuclear activity is the pacemaker for S-phase as well. We know it's needed for S-phase, and we know it phosphorylates substrates, but that's not the same thing as the pacemaker. So what we have to do is increase the activity in cells which have got an extended G1 and see if we push it earlier. We have preliminary work that is the case, and I think it will be the case, but I'm not confident enough to talk about it.
I mean, if you can change it, whether you can induce replication in a G2 phase.
I think we absolutely can, but in principle haven't done it, because if we kill the CDK and go down, we can then study what happens to the chromatin proteins during that transition from G2 to G1 to S. Do you remember?
Yeah.
So we started a chromatin project to look at that and other things.
And one more thing, the CDK, you said that export from the nucleus to cytoplasm because of permeability in the membrane. And when it goes to the next phase, how the CDK again goes back to the nucleus?
We haven't looked at exit from mitosis. So I can't, I mean, I know that cyclin is degraded. I know CDK activity in both cytoplasm and nucleus goes down together, but we haven't analyzed it.
Thank you.
Yes.
It's just a fangirl of yours since a very long time and wanted to attend your talk. So it was actually a very insightful talk. The question that I have is, we know that the substrates of the CDK are fluctuating throughout the cell cycle, right? And also, as per your talk, through the
When you say fluctuate, sorry, fluctuate in phosphorylation state or in protein level? Which did you mean?
The phosphorylation.
Yep. Okay, thank you.
Both, and from your talk, we get to know that it fluctuates in the nucleus as well as in cytoplasm. It's not the same. So my question is, when you say that only CDC2, CDK1,
Yeah.
is sufficient for the cell cycle to progress, right? So do you see this kind of fluctuation happening in the phosphorylation of the substrate throughout the cell cycle? Or does the phosphorylation status of substrates remain the same? And if it does change, how is the feedback loop regulated in these kind of cells when it is being governed by CDC?
I'm not quite sure what you're saying, but the regulation for sure occurs through tyrosine phosphorylation, but the mode of the regulation, by which I mean the strength of it, differs in the nucleus and the cytoplasm. It will depend primarily on the status of WEE1 and CC25, the balance there, which could be different in the nucleus compared with the cytoplasm, and I don't know the answer to that, and I don't know if that addresses partly what you're doing, and I have another answer if it didn't, which is that what this also implies is that there is a CDK regulatory domain in the cytoplasm and the nucleus, and by playing with nuclear exclusion signals and nuclear location signals, we should be able to create cells that are only going to activate the cytoplasmic CDK and not the nucleus, because eventually we will kick it up, or just nucleus. And that may divide the mitotic events, if I can put it in that way, into mitotic events within the nucleus and mitotic events within the cytoplasm, like growth stoppage and actin reorganization. So I don't know if that was addressing partly what you were thinking.
Part of this. And do the feedback loop of activation of CDC25C phosphatase regulated in this kind of cells when you have just a single CDK?
You know, something's a bit strange about that positive feedback, because it was shown in Xenopus probably 2 decades ago, and then nobody's sort of worked it out. So it was sort of speculated, decent data, but the sites weren't mapped. It wasn't sort of worked out, the careful dynamics. So I decided to go back and do that. We'd never done it in fission yeast before, so we were identifying the CDC25 sites. We can modify it and test this a bit more. I think what happened is, because this field moved incredibly rapidly about 30 to 20 years ago, I was around it, and people rushed on too quickly before actually defining it, and then they moved on to something else. So I think it's worth looking at it again.
If I may ask one more question. Thank you. The part where you spoke about the activity of CDK happening at the centrosome.
Yes.
There are various reports which have already suggested that they do see CDK activity at centrosome, which activates the nucleation of the spindle. So what do you propose here when you're saying that the analog with your CDK1, not CDK1, the CDK that you are studying does at the centrosome?
Well, and again, I think, and I'm not quite sure if I've got it right, I mean, I won't say it's the textbook view, but it is certainly a fairly dominant view that the centrosome is the pacemaker. And that's what you're referring to. And what I've just shown you is I don't think it is. In fact, we've almost shown it cannot be. At least in yeast, I mean, you'd have to look at it in mammalian cells. It's obviously playing an important sort of role, because we can't, phosphorylate substrates there, we don't get a mitosis. And I have wondered, 2 explanations. I mean, one is that maybe the spindle pole body or the centrosome plays some role in sort of ensuring it goes from the nucleus to the cytoplasm. But I actually now think that it does accumulate on the centrosome, but it won't do that if it doesn't get exported from the nucleus. So I'm only speculating there that maybe we should think about export rather than the association. This is just speculation at the moment.
Thank you.
But turn it on, please, and speak loudly. Tap it. No, not working. When you hand it on, don't switch it off, okay? On. Super.
It's not a question, actually, it's kind of a curiosity. So the project you're doing with the Sophie,
Yeah.
I see that nowadays that a lot of startups, they are doing the de-extinction projects. So is it kind of you're running in a parallel universe that you are de-extincting some kind of micro, at least a single cell organism?
No, I'm just doing Jurassic Park, that's all. Do you know, I find it an entertaining project. I can't pretend that it's, you know, pushing back the frontiers of knowledge, but I do think it's quite entertaining to predict what was happening and to reconstruct it. I tell you, I've never had any piece of my research ever reported in a newspaper, okay? And this is my last attempt to achieve it.
That's really interesting, thank you.
Of course, to say it's an excellent talk would be an understatement. It's not only the CDKs, but also the substrates of CDK.
Yeah.
If there is only one CDK that is important, then the repertoire of substrates is very small. How does the entire process work?
Right. So I think this is really worth thinking about. Many of these experiments have been done with about 300 phosphosites, which are on about 200 proteins. We now can map 450, something of that sort, and we're very critical about it. You know, we switch off the CD. It's not just consensus sequence. We switch off the CDK and look for exponential decay rapidly for time 0. So we got a single protein kinase hitting 300 proteins and 450 different phosphosites. Now, this is quite a demanding thing, I would say. And the reason I bring it up is that normally we think of phosphorylation sites as a certain consensus surrounding the threonine or serine in this case that gets phosphorylated. But if you are trying to regulate hundreds of proteins, there's going to be restraints on the sequence of those particular proteins, which to get a perfect or good consensus site may not be compatible with the other function it has to do, if you see what I mean. And this might not matter too much if you've only got a few substrates, but when you've got hundreds, it could be a problem. And we found the CKS protein. The CKS protein binds to the CDK and enhances the phosphorylation rate of about 200, and they are much enriched for weak consensus sites. So I actually think CKS might be a mechanism which can expand the repertoire of what can be phosphorylated beyond the immediate consensus site, which I think is the point you're making. Now, this is, I haven't written this up. It also does other things, by the way. It alters the regulation of WEE1 and CC25 too, because it can't associate. But I think CKS, which is in cancers, it's increasingly being shown that high-level expression is associated with cancer. And nobody's got any explanation for that, and I don't either. But if it's affecting CDK, then that could be a thing. Did I answer the question?
Yes, to me makes sense because of my interest in protein interactions. This might be the way that...
It's a way of escaping. Now, I'd never thought of that. It's not. We didn't think of it and tested it. It just was pondering about what it might mean.
Thank you.
Yes?
Yeah.
Oh, sorry.
Yeah. What I want to ask is that suppose you inhibit the activation in the nucleus, but in the cytoplasm, you enhance the activation. Will a reverse, like an import, can take place? Is it possible?
We haven't done specifically that experiment, although it would be an aspect of playing with an NLS and NES, playing around with that. And I'm quite interested in that to see whether, well, as I said, do we just get cytoplasmic events, for example, and nuclear ones, in those two different situations? But we haven't done that specific. But I think it could fall out, yeah.
Hello. Yeah. This is a simpler question among the rest. You, yeah. Hello.
Hello. Good. Just hold it 1 inch from your teeth.
Yeah.
Okay.
Hello.
Perfect.
So you mentioned getting suspicious whenever you read plasticity
Plasticity.
in a paper. Yes.
Get my gun out.
So at your level of experience, how do you like judge a good paper? How do you know that it's reliable? It's
Oh dear. Have you an hour? Well, I personally judge a good paper if I get excited by it. I mean, you know, and what I don't get excited by are papers which use a very clever technique. You know, the sort of thing the Broad Institute does. The Broad Institute, you know, they have these amazing technologies. They've got 30 authors. It's very clever, very difficult to do. You read all through the methods, it looks at thousands of things, and it concludes this much. Okay? I call it the disease, Broaditis, those sort of papers. Now, of course, I'm being rude, but I am a rude person, so you shouldn't worry too much about that. But I think you have to be excited. I think you have to have really good, convincing data, but also you need to have some ideas that are interesting, that are built on it. And I think there is a problem that we're having, if I can get a little more philosophical, which is this, is that our technologies are now so difficult that our poor students, postdocs and so on, spend all their time thinking about making the technique work. And they hardly have any time of thinking about what it means for the biology. And I have tremendous sympathy for them. And one of the things we can do, it's easier to do stuff in yeast. You know, it just is. You know, we've been doing gene editing since 1980. I first did it in the lab in 1980. And so we can do very precise experiments quickly. And so I try and give them time to think. I come back from here on Saturday, on Sunday, I take my lab away for a week with me, and we go to a retreat, and we don't talk, we talk about ideas. Not, I mean, with an experimental basis, but it's all about ideas. Because I just don't think we are spending enough time thinking about ideas. And a good paper should be addressing that. Something else. If you read The Origin of Species or something like that, Charles Darwin, you could still read it today and think this is something worth reading. I can barely read my own papers by the time they've gone through review, editorial, you know, anything interesting I might have to say is scrapped out because it's too speculative and stuff like that. And so, but you can read you can read 19th century and it's good. Yeah, it's good. Then one, two, whichever.
Can I ask one question?
Yeah, Dr. Serene and then yourself.
Okay.
Yeah.
So in 2002, when you gave an oration in this very auditorium.
Did I say something different?
No, you said something. You said something very profound after you were showing your papers and, you know, very nice papers. You said, on my one shoulder was angel, on my other shoulder.
Oh yeah, the devil. The devil.
And
yeah.
the context you used that you had very good data, you could have published in Nature or Cell or Science as you normally do. But there was something which was bothering you and you did not, you pursued that, and that led to very important discoveries.
Yeah.
So in the current context where scientists are judged annually,
I know.
The grant cycles also 3 years, at the most 5 years, PhDs have to be finished. So how do you do this? How do you suppress that devil?
Yeah. Yes, well, putting devils aside just for a moment, I mean, the point that you're making is really important. It puts a lot of responsibility on senior colleagues. They've got to do work at this. They don't just count what journal it's in, you know. I actually think Nature and Science, less so these days, so is a really, really good journals. You know, I respect them. But you shouldn't be but we know very good work doesn't get in there because it's a sort of, you know, random whether it works or whether it doesn't. I mean, you know, it really is. Like, I mean, it's done for a lot. But when we are judging people, we have to read the papers. We have to read the papers. And the whole system is biased a bit. I mean, it's probably more difficult getting something published from India and China, for example, because everything's sort of built around the US mostly. It's even a problem a bit more in Europe. And so getting into high-profile journals is not, it means something, don't eliminate it, but you have to read the papers. And I think senior colleagues are sometimes lazy and don't do it. And I think that's and it's the same with granting as well. And what's made worse with granting is that no, I mustn't say this. Those who work in the research councils and so on, they get funny ideas about what works, what doesn't work, and whatever. So the Wellcome Trust, for example, has just said we're not going to look at any work that is previously published to make a judgment on quality of the work. Now that's just lunacy, you know. It's complete lunacy. But what they're trying to say is we have to have a level playing field, as if what we write in our grants we're intending to do anyway. I mean, it's actually written just to get some money in the place and then go back to nobody seems to all. But you see what I'm saying? You have to write a grant in a particular way to get the money. I mean, I'm a practical person, but it is barking mad half the time. Yeah.
You know, the point I was trying to get from you is that you had the data you could have published, but you did not go.
Correct.
That you wanted to get to the bottom of it.
It's correct.
And that's what. So you could have still got a very good paper. Of course, your standard is Nature and Cell and Science, but you can't beat that. So where do you see that when people have got something which is good, publishable, but they want to go more into it because something they are not understanding?
Well, I think you have to be honest with yourself, and you do it, you know, and in the end it will come out right. Yeah. In the end, you've got to follow your principles.
Thank you, sir, for the wonderful talk. And what makes it so memorable is the infusion of humor into your talk. So we really kind of enjoyed the talk. So my question is, like, in one of your graphs you have showed the CTK activity kind of linked with the cell length.
Yes, yes.
Right. So my question is that did you find in your studies any kind of interaction with the cell division protein of the CDK?
I think the cell length thing is that you spotted is critical, because cell length is of course a reflection of cell size. And what is restraining in this system, the pacemaker, is the size of the cell. How does the cell measure its size? And I still don't know. And I mean, I remember asking that question in 1975. So that's now is 50 years ago. I thought we'd solved it in about 2008 and then did another experiment and we didn't. Okay. And I think it's a really interesting thing. So the length is a reflection of size. And this is a fundamental property. You have to measure how big you are before you activate. Now, it's got to be something to do with cycling, CDK, WE1, CC25. And I mean, we know all of these things influence it. And I've got I've got 6 students, I said, and one of them is just looking at heterozygous deletes of all these combinations, because you have to do what I call local perturbation experiments. If you hit the gene too hard, you'll get you take the system out of its control space, because you're working within a sort of parameter. You delete a gene and you're out here, and so what you discover out here may or may not be significant to what actually is happening. But if you do experiments where you only modify the levels a limited amount, so you go down 30-40%, go up 30-40%, you're more likely to remain in the control system. So, for example, people have measured CDC25. They found when cells get bigger, it goes up. So they say it measures cell size. Okay. But if you change the level of CDC25 50%, it only has a 5% effect. So it's simply not true. And it's unsolved. And I'd like to solve it before I die. I really would like to do that, because I think it's interesting and important.
So I think there are no further questions, Professor Nurse. So we'll hand over the mic to our Master of Ceremonies. Thank you very much.
Thank you.